need to check the parasitemia, because if there is too much parasite in the red blood cells, the parasite is going to die. So I take the flask, I gently swirl, try to avoid any bubbles. There. I have to detach the red blood cells. Alright. So I tilt it, try to avoid to touch the rim. Now pipette inside. Okay. Normally it's already set up. There is a tube there to balance. RPM for 3 minutes. There's only 2% of red blood cells in the media, right? And so I spin it down, and here you have the infected red blood cells at the bottom here. Just breathe up until the layer there. Okay. Then you mix a bit. Take a bit of the parasites, put it there. And make a smear. Right, and you can see like you have like the parasite everywhere. There it is. There. One. The goal is to put Giemsa everywhere. Right. I'm gonna do that for you. Alright. Of course. I will zoom it in, but if you want to take a look. I think sure. So here you have got l'Agitating, so the red blood cells are ready to burst. Here you've got all of that, you've got like ring stages, so it's pretty early. It's like 12 hours post-infection. And here we've got like trough. Right, so like you can see, like it's multinucleated. There is what we call Amazon crystals. So that's what. So basically, what's staying here is Amazon crystal, right? Because the parasite is going to digest hemoglobin, and so the hemoglobin contains iron, and iron can induce like very strong oxidative stress to the cell. So the parasite needs to find a way to take the hemoglobin, take the heme molecules and make it inactive, so it doesn't create like oxygen radicals. So to do this, it's gonna polymerize the heme molecules, and it's gonna make like these dark crystals that you can stain on the like with the Giemsa. 500 RPM actually. 4 or 5 minutes, and yeah, temperature is fine. And that's it. So here is the blood. Cool. Here you have the plasma. At the interface here you have the buffy coat layer, which is white blood cells, and there you have the RBCs. Now you need to aspirate, and you want to aspirate the buffy coat. And then some bleach. Got it. Then I will have 50% red blood cells. Right? Right. And then, so you have this. Oop. Right. Oops. From it and put it there. So you want to make sure you detach the red blood cell. So an homogeneous solution, but you want to avoid bubbles.
Always make sure it's balanced.
Okay, so my name is Dimitri Desmonts-Delamache. I'm a PhD student here working on Plasmodium falciparum metabolism, and more precisely on the digestion of hemoglobin by the parasites Plasmodium falciparum. So what's important when you work with parasites or even viruses and prokaryotes, that it's better to avoid like mixing them together, so having them in the same space, because like you can have cross contamination, and this will have an important impact on the data that you will get at the end of your experiment, right? So if you work with prokaryotes and someone, or like you have someone didn't clean the bench properly, or you have some leaking or whatever, you might create this space, especially like with cells that can be infected by the bacteria. You can have this very big spread and contamination, and then it's gonna skew the data, and you might not know why, because when you do your experiment, normally you shouldn't test all the time against every possible bacteria or every possible pathogens, right? So maybe you will see something atypic and you'll be like, oh, there is a phenotype that's new that we never saw, but it's actually because there is this contamination that was there in the first place, and you didn't test for it because it shouldn't be there. So if you have separate rooms, it limits the risk contamination. Basically, to synchronize the parasite, we put sorbitol, which is a sugar, and it's gonna select, or actually only parasite at a certain stage can live in sorbitol, so the other ones are going to die. And this is specifically the ring stage parasites, and so this is very important because metabolism evolves during the life cycle of the parasite. So basically, if you have asynchronous parasites, you might have tons and tons of various metabolites that are not necessarily important to you, right? And especially for me, I'm studying the parasites at how the parasite digests hemoglobin, and the digestion of hemoglobin is going to be achieved, or like hemoglobin is going to be digested at 80% at the end of the life cycle. So if I have all the parasites at that are synchronous, I know that at this specific time point, most of the hemoglobin will be digested, and so I would have also a better signal, right? So yes, you want to avoid to have like too many metabolites that are due to life cycles that you might not necessarily, to life stages that you might not necessarily care about, and also it's to increase the signals at the specific stages that you care about, right? Because you know that, okay, tomorrow at noon, all my parasites will be ready and at this stage, and now I can do the experiment because this is the stage of interest, right?